This is a production of Cornell University.
Okay. Hi, everyone. Sorry for the delay. Today, I'm going to talk about the benzoxazinoid pathway. And there's a lot of genetic redundancy in the pathway. And so I'm interested in like why that might be. So why are benzoxazinoids important for plants? These are the various compounds with their decorations down in the lower right. Many compounds from a common precursor. So these can provide resistance to biotic stresses such as insects, nematodes, microbial and fungal pathogens, for example, northern leaf blight. Also, they've been implicated in, providing tolerance to abiotic stress. They can chelate metals, and as we heard about 7 weeks ago from Jiping, there's evidence that they can provide protection from plants from aluminum toxicity. There's also evidence that benzoxazinoids are involved with allelopathy. They're phytotoxic, including autophytotoxic, and I'll talk about that in a second, because root exudates can be taken up by neighboring plants and harm those plants. There's also evidence that benzoxazinoids are involved with regulation and signaling, including hormonal, transcriptional, post-translational modification, and can also control other secondary metabolites. So here's the core benzoxazinoid pathway. The benzoxazinoid pathway is not found in all grasses, but it is found in maize, wheat, rye, wild barley, and some other random dicot plants thought to be a result of convergent evolution. So the pathway here is where it stops in rye and wild barley. But in the predominant benzoxazinoid in maize and wheat is this molecule called dimboa, which requires 2 extra steps with, that 2 other genes control, BX6 and BX7, which are not shown here. So this is the core pathway. What you can see right away at the top is laser pointer is there's 3 copies of IGPS1. So this is indole 3-glycerol phosphate synthase. My lab mate Annette Richter just published a paper this month in Plant J that has showed that this channeling that you see between these 3 branches down below actually starts upstream here. So she's just shown some good evidence for that. But what you can see is the pathway does this common IgP3 precursor branches off into the tryptophan pathway, which is essential for being alive in plants. You need tryptophan, and auxin is downstream of that. And then the indole provided by this BX1 gene feeds into the benzoxazinoid pathway. And then this other gene, both of these derived from tryptophan synthase A, evolutionarily leads to the indole being converted into volatile indoles, which generally either attract parasitic insects or repel herbivores. So obviously I'm going to focus on the benzoxazinoid pathway here in the middle. The thing I want to draw your attention to right now are 2 parts of this branch of the pathway where BX1 converts IGP into indole, and then the indole is subsequently converted, it's oxygenated, monooxygenated by a cytochrome P450 BX2, okay? So, and the other part of the pathway I'm gonna focus on is this part here that's controlled by BX8 and BX9, which accept the same substrate, diboa, in this diagram, but it also will accept dimboa as substrates. So BX8 and BX9 are highly redundant in their function in glycosylating the diboa molecule, which prevents autotoxicity. So this sugar moiety is added to the molecule, and then eventually it's transported into vacuole as a phytoanticipin for herbivory, where glucosidase will cleave off this sugar molecule, and then the toxicity of the diaboa molecule is restored. So as far as BX8 and BX9 go, what's the reason for the genetic redundancy? BX8 is induced by herbivory at much higher levels than BX9, based on some previous work in our lab. So I'm curious as to what's really is the primary function of each enzyme. Some possibilities are that it could be strictly, they just do the same thing, benzoxazinoid biosynthesis. It could be that this allows transport across the tonoplast membrane, as in anthocyanins, this is required. It could be that one of the molecules or both provide detoxification of the A glucones after herbivory, so it doesn't harm the plant any further. And it could be that there's some developmental or cellular subfunctionalization or compartmentalization involved. So I have two project goals for this, and one is to contrast the role of indole versus benzoxazinoid signaling, because they can both act as signaling molecules toward insect defense. So what I plan to do is break the benzoxazinoid pathway both before and after that indole biosynthesis step between BX1 and BX2. So I decided to take a targeted mutagenesis approach, since there was no BX8 mutants available, to create, sorry, this is BX1, BX2. I was going to create a BX1, BX2 double mutant to try and tease this apart. And then I'll compare the double mutants versus the single mutants versus the wild type in a series of metabolic profiling and insect bioassay experiments. For the BX8, BX9 portion of this project, I want to determine the level of functional redundancy between BX8 and BX9 by conducting a targeted mutagenesis of BX8 and verification of available BX9 mutator alleles, another transposon which can inactivate genes. Then I'll cross the BX8 and BX9 mutants, compare double to single to wild type in a series of metabolic profiling and insect bioassay experiments. So today, this is gotten as far as I have the genetics, and I'm about to embark on the metabolite characterization, metabolic profiling of the benzoxazinoids in my mutants. So most of the benzoxazinoid genes are clustered in bin 4.01 in maize. And you can see that BX1 and BX2 are very close together, about 3 kb apart. Most of the cluster resides in this 264 kb region. And BX8 is about 42 kb upstream of BX1. This is all in the W22 inbred. So I have a BX1 DS donor, which was isolated many years ago from a previous project I was involved with. And the idea is to jump BX1 into BX2 through transposition and do a co-screen looking for insertions in BX8. So the idea is to knock out these two genes independently, thereby creating the mutants I'm after. So I won't go through this is published. I'm not going to go through the gory details of the genetic scheme, how I built my population. Basically, I have a line that has my DS donor in BX1 with an AC source in the background to provide the transposase that the DS needs to move, test cross it, and then I get an ear looking like this. Each kernel is a potential independent transposition event. So then we grow out all the progeny from the ear and make pools of seedlings at about 10 to 14 days or so. And so lately we've been pooling about 18 seedlings in a single DNA prep for the purpose of the PCR reaction. And using, by multiplexing gene and DSN primers, I can pick an event in one of my target genes if a DS happens to jump into that gene. So thanks to my awesome interns that have been involved with this project, both Rawlings scholars. Serena Lotrek was a former intern, now in grad school at Michigan State, and my current intern, Abby Kelly. So Serena was able to create 2 double isolate two double BX1, BX2 double mutants, which we call 1514 and 1816, arbitrary names. One, the DS donor was not retained, so I have an excision footprint allele here and an intronic insertion here. So I would expect 1514 to not be a true double mutant, in that this DS should get spliced out during transcription. The 1816 mutant is a little more interesting in that it retained the DS donor in BX2 in this case, and it inserted two base pairs downstream of the exon 4 intron 4 junction. And I won't show the data here today, but what this caused splicing here. So I'm getting a chimeric transcript of wild type and mutated BX1 transcripts. So what I think that will do is it'll probably it'll produce some indole, but not a whole lot of indole. Abby was able to isolate, she jumped the other way from a BX1 donor into BX2 and was able to isolate 3 alleles. 2 of them retained the DS donor, and one of them lost the DS donor, left an excision footprint. But all her insertions in the target gene BX2 were exonic, so that's good news. Unfortunately, we lost 2 of those alleles because we couldn't recover viable seed in the greenhouse. So that's the breaks. However, we do have a really good BX1, BX2 double mutant. I've just gotten a homozygosity in the greenhouse. That's ready to harvest next week. So I'm going to dry that down, and then I can jump right into metabolite profiling. So Abby was also able to isolate 3 BX8 DS mutants. We jumped both from BX1 into BX8, BX2 into BX8 because we had those populations. And she was able to create an exonic double mutant in the 13.5 allele. We have an excision footprint in the 38.14. And then this one was upstream 344 base pairs upstream of the translation start site in BX8. So I'm not sure whether 7.8 will have any real effect or not. We'll just have to wait and see. Unfortunately, we lost, so 13.5 and the 38.14 insertion sites were identical. I know they're not a contaminant or something because one retained the DS donor and one did not. They're independent. So this could be an insertion hotspot in the gene, but unfortunately we lost the 38.14 allele and only have the 13.5 allele to work with, which is fine. Okay, so I identified 2 uniform mu insertions in BX9, which I wanted to cross to the BX8 allele to make the double mutant. However, one of the stocks was unavailable. The other stock was sent to me from the Maize Genetics Co-op, and I couldn't confirm the insertion 0 out of 15. So that left me kind of stuck until there was a new transposon resource that emerged at the end of last summer. This on-mute collection that was developed by the Hochholder lab in Germany. So I looked at BX8 and BX9 insertions, and I found 3 in BX8, 2 are in the 3' UTR, so I didn't bother even looking at them, and decided to focus on the exonic insertion in BX8. Luckily, I had 1 out of 10 mu positive plants, which I was able to recover and propagate. As for BX9, there were a lot more bond mu insertions. I decided to focus on, again, 5 intronic insertions. 2 of them dropped out. I couldn't confirm them. So that left me with 3 exonic insertions. Again, this is fine. I was able to identify several plants that verified in a PCR assay as containing these bond mu insertions. So that's good news. So what this means is I decided to use my BX8 bond mu as a pollen donor onto these 3 BX9 alleles. I made those crosses just last weekend, so they're growing up in the greenhouse right now, and I should have a really good BX8, BX9 double knockout, as well as the BX1, BX2 knockout to characterize. And that's all I have for you today. So I'd like to thank the organizers for talking me through my power failure and for putting all this together. And also the student speakers, you always impress me with the work that you're doing. So I thank you for giving me more insight into what you're doing. My committee, George, Margaret, and Michael, thank you. SIPs, TA funding, I couldn't do this without you. I don't have any money. So thank you, Carlin, Michael, and Bob for the TA ships. Greenhouse staff and the other Jander lab members, which has made this COVID experience somewhat normal. So thanks for the discussions and the help with my work. Thanks, and I'll take any questions you have.
Thank you, Kevin. That's a wonderful talk, and thanks for thanking all these people. That's really good. You're right.
I have a question. So you had a lot of trouble recovering viable seed. Does this pathway feed into other pathways that are necessary for the plant that you know of, or is that unknown, but likely based off of what you found?
I mean, I'm quite sure that it's involved at this point. It's involved a lot of different pathways. The evidence keeps stacking up, and there was a really nice review I was just made aware of by Matthias Erb and Dan Klebenstein that came out in 2020, which really sort of characterizes all the things that benzoxazinoids might be doing in addition to defense. It's a lot. So, but I think as far as not being able to recover viable seed, I think it's more the struggle with the greenhouse conditions. Sometimes I get good pollen shed, sometimes I don't get good pollen shed on the exact same genotypes. Sometimes plants are nicking at the same time. So I think it's more of a practical function rather than a biological function for the loss of the alleles.
Okay, great. Thanks for the question and nice talk.
Thanks, Savannah.
I can't see any other questions in the chat, but I also have a question for you. So when you want to look at the BX8 and BX9 complementing each other potentially, so you're only looking at these two and not at other BX genes, right?
Yes.
Okay. And how, I'm just wondering, like when you knock both out, how potentially, or if you knock one of the two genes out, how potentially like RNA silencing, like the cell's response to not having one of the genes anymore. How are you, or are you taking that into consideration?
That's a great question. I don't know how RNAi might affect this. All I know is that the benzoxazinoid molecule, when it's not glycosylated, is toxic to the plant, right? And so what I'm wondering is, if you knock out one of the genes, BX9, and have BX8, or vice versa, you have BX8 but not BX9, can you survive, right? I think you probably can, but if you knock both out, is the benzoxazinoid toxic enough to kill the plant? And I don't know that until I can see the double mutants. One thing I didn't show here is there is another glucosyl transferase that's been implicated through network analysis that was previously not shown to be involved with the benzoxazinoid pathway. Since these are promiscuous enzymes, I'm wondering if this sort of third benzoxazinoid glucosyl transferase, which has a relatively high homology to BX8 and BX9 at the amino acid level, can serve that function and will rescue the double mutant if it would be toxic. So
yeah, it sounds like something you'll see at the point when you're having the plants. That's very interesting.
Yeah, that's all I can do, but I at least have the double mutants in hand now, so I'm on my way.
Yeah, great. I'm glad. Well, thank you, Kevin. I don't see any other questions still. For everyone that's still here, please remember to fill out the feedback forms if you haven't done so. This was the last talk for this academic year, and we'll be happy to have talks again next year. Thanks everyone for attending, for asking questions, and for filling out the feedback forms. And have a good weekend.
Everyone.
This has been a production of Cornell University, on the web at cornell.edu.